Good afternoon, students. Welcome to the second part of this practical session in Dotmatcher. So, we need any DNA, RNA, or protein sequences to execute the Dotplot experiment. So, I'm going to download this from Uniprot, right? You already knew that if you want a gene, you can go for NCBI, and if you want a protein sequence, you can get it from the Uniprot, right? So, Uniprot, I already have. Just you can go for hemoglobin for my example. So I get so many entries. So I need human, right? So I will get the human sequences here, and also you should see that whether it is reviewed reference sequences. Okay, so you can just click here. You can find the download option. So just click it. You can download. I already downloaded, so I just show you. And next I need mouse hemoglobin. So hemoglobin beta, right? For mouse 147 amino acid, that also a reviewed Swiss prot reviewed sequence. So just I can click here and I can download it, right? And now I can go for what is that? I can go for zebrafish, right? So it is again Danio Rio. So I can just click and I can download it. Okay, so by this way you can download three individual sequences uh, to compare. Okay, in the to going to align by using Dotplat. Okay, so I have already downloaded. Just I check it whether it is in the download or not. Okay, and this is the downloaded file. I want to know that this is Danio Rio, which means zebrafish. So I will rename zebrafish amino acid sequence. Okay, so likewise this will be in a rat mouse, right? Mus musculus. So this will be an albino then. So I will rename that albino HV. Okay, amino acid. Sure. Here also I put that, so that is hemoglobin sequence HV amino acid. Okay, these two I should have the files. Best I will get here. So already I have zebrafish hemoglobin amino acid anyway. So we will get skip. Okay. So let's check that whether it is okay. Then now I will go to sequences are ready. Now we can go for EMBOSS Dotmatcher, right? So I will go for EMBOSS Dotmatcher. See that you can find the sequence type, which sequence you are going to give as an input. So if you are going to do it a protein, you can just click it. Likewise, DNA means you just click it. And if you want, you can copy and paste the sequences. And just because I have the file, just now we downloaded, so I have just insert the file. So I'm going to work in the protein. So I'm going to upload the amino acid sequence of the particular protein. So two proteins we need. So the first sequence is maybe where it is from. It is from sequence analysis. It is in files. Okay. The files first I want to compare human amino acid, human hemoglobin beta subunit, right? With the second sequence will be the mice, albino mice, albino, what it is, albino hemoglobin amino acids. That is a beta subunit. Okay. So I have already loaded the two sequences. If you want, you can just copy and paste here the sequence, right? So if you paste here, you will get the name also. So it is better you can and give the sequence, just copy and paste the sequence, right? So more option, you have just click this. See, this is Blossom 62. You can use 3540 also. Window size is 10. So I already, the sequence is nearly 150 and 167, I think, the length of the sequence. So it is better for 10. I leave as a default. If you have the lengthy sequence means you can just increase that window size and threshold level also, right? Okay. If you want, you can give the title. So this about human versus albino or mice. Okay, I can just submit it. Once the job is over, you will get the prompt here. So job is ready, the result is ready like that. So you can click, see that. So job is get finished. Leave the result and just click here, and it will take time to load the plot, right? So like this way you should perform these two also. Zebrafish versus human, right? So then zebrafish versus albino. So you will get plots, three individual plots. So it is. So if you take time means you can just go here. You can download the tool output, right? So by downloading this, you can get the plots. We'll wait for some time. It is getting loaded or not. Okay. So like this you should perform all the three, and you should analyze these things, whether how many diagonal lines are there, which means whether there is a continuous or disruptions or anti-diagonal is there, means the main thing you should know is if you have more noise means you should increase the threshold value and window size, right? This is what, see that. Unfortunately, 100% match has been noted here. So this is hemoglobin human and this is hemoglobin mouse. This is the first, what is that, first amino acid to more than 100 amino acid, maybe 147, I think, 147, 150 amino acids are there. So we have, there is no dots other than this diagonal line. There is no parallel line, there is no anti-diagonal, there is no cross, and there is no little bit dotted is here the both side, right? So you may get for your results you may get so many dots here and there. So for that you can easily, in my case there is no dots or diagonal lines, small lines are not available here. So I will show you how it will be in the as an example. See that you can just dot plot. You can go for images. Where is that? Okay, dot plot results. No, these are not exact. I will show you the EMBOSS dot plot. Okay, you can see here so many examples, and this is what exactly happening, right? In this, you can see so many dots are there, and here you can find there are three. Shall I click here? We will show that. We will see that. Okay, we'll go to the research. Okay, in the meantime, we'll see. Here the next example. See that the plot is in this direction, so 0 should be here, right? 0 and 1 2 3 like that, 0 1 2 3 like that, so diagonal will be in this side. In our case 0 will be here, and 0 starts from here, so the diagonal will be like that. Anyway it should be in the right angle, right? You see here there is a continuous diagonal and there is a gap here in this image, right? Gap, that means there is a mismatch or deletion is happening. You see that there is a parallel diagonal line, right? In this example, the diagonal will be in this in for our in our table or in our plot, the diagonal will be in this right angle, right? Here, because of the 0 is here, the alignment sequence is put from here to here, and second sequence will be from here to here. That's why the direction of the diagonal is changed. Okay, so the parallel means you can go and refer that table. What I have told you about the parallel means it repeats. Okay, parallel find, see that some bases where some amino acids or bases or sequences has matched here, and there is a gap, and there is already the another match is here like that. See that, and the last end of the first sequence is matched with the beginning of the second sequence. So like that you can find. So a little bit local similarity is here. So like that you can find that. See that so many dots. If you get a result like this, this means this is so many noises are there. So you have to increase your threshold level, right? Threshold level and window size. That's all about the examples. See that this also so many, because of the less threshold value and less window size, you can get so many matches and this will be interfere. We cannot find the console agents. Okay, so I will show you in this example. You can see that there is a anti-diagonal. This is a diagonal, and this much similarities are there, but some gap also present. So this amino acid are, if the base means one base, and this will be an another insert deletion is happened or missimilarity. And here see that there is a gap in between and this base up to this one. And what happens, there is a line coming as an anti-diagonal, right? So you should know that what it denotes. These are all things you have to discuss in the result. And use the so many noises are there in this picture. Okay, this is about, so see that exact example with the optimum uh, threshold. They have, like us, they have given the three samples, so all are in the diagonal. In that you can find, you can read this also, right? On the two frame shift, see that in the first picture there is a perfect diagonal line with the two frame shift mutation sides, with the two frame shift mutation lines, right? The second one revealed low complexity region between domain. Second one, low complexity means there is an there is many lines, which means there is no other lines. A lot of lines are not available here. So here, little bit available means there's a complexity. This is very less complex. And here in the third one, you can show that two areas of different amino acids. Show two areas of different amino acids are there in that. See that how do you say that two areas of different amino acids will be present in that means there will be the first area and then the second area. I think I hope if you not understand means you just leave it. We will see in the another software. So I can explain you by using the graph with basis here. We will take a simple example by using uh, shortest length and we will explain. I will explain to you. Okay. So this is how you can explain discuss the result whatever you get for your sample, right? So what is that? For this. So this is all about the dot plot. So like that, you have to go for human versus albino here, and human versus zebrafish, and zebrafish versus human. Sorry, zebrafish versus albino. For any proteins, I have taken here as the hemoglobin. For you, you can take your own gene of interest. You can compare with any species and you can submit the result. But you, I told you, don't forget if you get any more spreads, which means more amount of dots here and there and lines, shortest lines, repeats, means you can increase the threshold value. See that? You can go, you can submit, you can increase, just click this and you can put as 25 or 20 like that. Here threshold value you can go for up to 100. Okay, and this is about the embossed Dotmatcher. Hope you can understand everything. You can do that by today. Experiment and submit it. Okay, thank you.